If you look at a living thing under a microscope, you generally expect chaos. Right?
Oh, absolutely. Just a frenzied soup.
Yeah, exactly. We imagine our cells just like constantly shifting and splitting and colliding. But what if that chaos is actually an illusion?
It totally is.
Right. What if what you are really looking at is this highly regulated, tightly controlled, literally life or death security system? So today we are doing a deep dive into a huge stack of research entirely focused on the cell cycle.
And this is so crucial.
It really is. Whether you are, cramming for your TEAS or HESI nursing entrance exams, or if you're just someone who's insanely curious about the hidden machinery keeping you alive, our mission today is to completely dismantle how you look at microscopic life.
We really want to get into the how and the why here.
Right. Like, why do your skin cells heal in a few days, but a spinal cord injury is permanent? Or how do chemotherapy drugs actually work? The answers to all of that are hidden in this one continuous cycle.
Yeah, it's the underlying rhythm of all tissue growth and, well, all disease, too. I mean, if you don't understand the sequence of events a cell uses to duplicate its contents and divide, you simply cannot understand how a human body functions.
Or how it fails.
Exactly. How it fails.
So I want to start by addressing a term that basically everyone learns in high school biology: mitosis.
Oh, right, the classic trap.
Yeah, because for the longest time, I thought mitosis and the cell cycle were completely interchangeable terms. And looking at the research, it seems like most students and even 19th century scientists make that exact same mistake.
They do. It is probably the most common pitfall on any clinical biology exam.
Why is that such a dangerous trap, though? Like for someone trying to really understand cellular biology?
Well, it really comes down to a fundamental misunderstanding of time and effort. The cell cycle is the complete start-to-finish sequence. It's everything a cell undergoes to duplicate its genetic material and physically divide into two daughter cells.
Okay, so where does mitosis fit in?
Mitosis is just a tiny, fleeting fraction of that whole process. Specifically, it's only the division of the nucleus.
Just the nucleus.
Right. It's the flashy, dramatic finale where the chromosomes condense, dance around the center of the cell, and get pulled apart.
The fireworks show.
Yeah, exactly. But the heavy lifting, the actual reading of genes, building of proteins, copying billions of DNA-based pairs.
The actual work.
Yes.
Yeah.
All of that happens entirely in the shadows, long before mitosis even begins.
Which means we are missing the vast majority of a cell's life if we only focus on the division part.
Vast majority. Yeah.
So how did we end up with such a skewed view in the first place? I mean, that seems like a pretty glaring blind spot for the scientific community to hold on to for decades.
You have to put yourself in the shoes of a scientist working in 1882.
Okay, 1882.
There was this German anatomist named Walther Flemming. He was peering through an early microscope at the tissue of a salamander.
A salamander?
Yeah, because he had developed these new ways to stain cells.
Yeah.
And he was the very first person to actually see these thick, thread-like structures.
The chromosomes.
Right, the chromosomes.
Yeah.
He saw them condense and pull apart because he could physically see this incredible microscopic choreography. He coined the term mitosis.
From the Greek word for thread, right.
You got it.
Okay, so that makes sense. He named what he could physically see happening, but what was the cell doing the rest of the time under his microscope?
Absolutely nothing visible.
Nothing.
Between one round of mitosis and the next, there's just this long, apparently featureless interval. Under a 19th century microscope, the cell just sat there looking like a stagnant, watery blob.
Just a blob.
Yeah, and because of that limitation, scientists labeled this massive block of time interphase. They literally defined it as the resting phase.
The resting phase.
And that assumption essentially closed the book on the subject. I mean, for roughly 70 years, interphase was a complete black box.
That is wild. Calling interphase a resting phase is, it's like calling the 9 months of human pregnancy a resting phase just because the actual birth is the dramatic, visible event at the end.
Oh, that is a brilliant way to frame it.
Right. Because it completely ignores the staggering amount of biosynthesis and, the genetic replication and nutrient gathering happening behind the scenes.
Exactly. The resting label wasn't a description of reality. It was just an artifact of bad technology.
Wow.
Generations of biologists just assumed the cell was asleep, waiting to divide again.
So who finally shattered that assumption? Because someone must have figured out a way to look closer.
2 British scientists finally cracked it open in 1953. Alma Howard and Stephen Pelk.
Okay, Howard Pell.
But the funny thing is, they weren't even trying to map the cell cycle out of pure biological curiosity.
Really? What were they doing?
They were working at the Hammersmith Hospital in London, and they were tasked with studying how ionizing radiation damages plant tissues.
Oh, right. The research notes mentioned they were using bean root tips for this, the Vicia faba plant.
Yes, Vicia faba.
But how do you use radiation to look inside a cell's timeline? Like, how does that work?
So they used a technique involving a radioactive isotope called phosphorus-32. Now, DNA is absolutely packed with phosphorus.
Right, the phosphate backbone.
Exactly.
Okay.
So by introducing this radioactive phosphorus to the bean roots, they could literally track the glow of the radioactivity.
Oh, wow.
Yeah, they could see exactly when the plant cells were actively building new DNA. They were hunting for the exact moment of synthesis.
Let me guess. The glowing wasn't happening all the time.
Not even close.
Yeah.
The DNA replication wasn't continuous throughout that long resting phase. And it absolutely wasn't happening during the flashy mitosis phase.
So when was it?
The glowing radioactivity was confined to one highly specific discrete window right in the dead center of the black box.
Okay.
Howard and Pelk named this window the synthesis phase, or S phase.
And because this synthesis window was isolated in the middle of interphase, there had to be like a gap of time before it started and another gap of time after it finished.
Precisely. They labeled the gap before DNA replication gap 1, or G1.
G1, got it.
And they labeled the gap after replication gap 2, or G2.
Okay, that makes so much sense.
Right. And just like that, they shattered the myth of the resting phase. They birthed the modern four-letter vocabulary, G1, S, G2, and M, that forms the backbone of cellular biology today.
I want to translate those 4 letters into something tangible for you listening. Let's walk through a cell's life cycle chronologically, using a standard 24-hour human cultured cell as our baseline.
Sounds good.
So the newly divided cell has just been born. It enters G1. What is actually happening in this first gap?
G1 is incredibly active. The cell is physically growing in size. It's synthesizing mRNA and protein.
Getting bigger.
Yeah, and it's multiplying its organelles, like the mitochondria, because it has to eventually support 2 whole cells. But more than anything, G1 is a decision room.
A diffusion room.
Yeah. The cell is constantly sensing its environment, like, are there enough nutrients around? Is the temperature right? Are surrounding cells sending chemical growth signals telling it to divide?
Is its own DNA completely free of damage?
Exactly. It's taking a massive inventory before committing to anything.
And in 1974, a cancer researcher named Arthur Pardee discovered that there's a specific physical point of no return late in this G1 phase.
Yes, the restriction point, or the R point.
The R point.
If a mammalian cell gets the green light to pass the R point, it no longer needs external growth signals. It is biologically committed to completing the entire cycle, up to and including dividing.
Wait, if the R point is the point of no return, what happens to the cells that fail the inventory check? Like, what if there aren't enough nutrients and the body just doesn't need any more cells in that area? Do they just, die?
Usually no. If growth factors are withdrawn before the cell hits the R point, it shifts gears. It exits the active cell cycle entirely and parks itself in a specialized state called G0.
G0. Okay. I am looking at the clinical applications of this G0 state, and this feels like one of the most vital concepts for anyone going into nursing or medicine.
Oh, it absolutely is.
This isn't just trivia. This is the reality of severe trauma.
It's the exact reason why certain catastrophic injuries are permanent.
Because of G0.
Right. Mature neurons in your brain, skeletal muscle cells, and adult cardiac muscle cells.
The cells that make up your heart.
Exactly. They do not regularly divide. Once they mature, they exit the cycle and park indefinitely in G0.
Wow.
That is why a massive heart attack or a severe stroke leaves permanent scar tissue. The surrounding heart or brain cells are locked in G0. They cannot re-enter G1 to multiply and replace the dead tissue.
Are they permanently locked, or is it just like a really deep sleep? Because if G0 is just a dormant state, couldn't medicine eventually figure out a chemical signal to wake them up?
That is one of the most heavily debated frontiers in biology today.
Really?
Yeah. Is G0 a reversible state of quiescence, where the cell retains the ability to divide but just chooses not to? Or is it a state of terminal senescence, where the machinery is basically permanently dismantled?
Because we know stem cells can drift in and out of G0 as needed.
Right. If we could ever figure out how to safely force a damaged heart to coax its surviving cells out of G0 and back past the R point, we could theoretically cure heart disease.
That is incredible.
It is, but the risks of doing that are astronomical, which we'll get to a bit later.
It's staggering to think about, but let's follow the cell that does get the green light. It passes the R point and moves into the S phase, the synthesis phase that Howard and Pelk discovered with the glowing phosphorus.
Right.
In our 24-hour cell, S phase takes about 8 hours. I'm trying to visualize this. A human cell has to copy roughly 3 billion base pairs of DNA.
3 billion. Yeah.
Doing that in 8 hours is like sitting down and copying an entire multi-volume encyclopedia by hand, letter by letter, in a single afternoon. How does the cell pull that off without introducing massive, catastrophic typos?
Well, it manages that speed because it doesn't just start at page 1 and read to the end. The cell uses thousands of replication origins simultaneously.
Oh, okay.
Imagine thousands of scribes all starting at different chapters of that encyclopedia at the exact same time, working until their sections meet.
That makes a lot more sense.
But you're entirely correct to worry about typos. The speed absolutely invites errors. The wrong genetic letter gets inserted, or a strand physically breaks.
Which is why the cell can't just immediately jump into dividing. It needs time to proofread the encyclopedia.
Exactly. And that brings us to G2. Gap 2 takes roughly 4 hours in our 24-hour model. G2 is a mandatory quality control stage.
So it pauses.
Yeah, the cell physically pauses to double-check that every single strand of DNA finished replicating and that the sequences are intact. Specialized proteins run along the DNA, hunting for structural damage and repairing it before the cell is allowed to physically separate the chromosomes.
Okay, so the cell has grown and gathered nutrients in G1, copied the entire genetic code in S phase, and proofread that code in G2. That entire sprawling, complex process was interphase.
The invisible work.
Right, the invisible work. Finally, after all that, we reached the M phase, the part Walter Fleming actually saw.
And the M phase is shockingly brief.
Yeah.
Usually just 1 hour out of the entire 24.
Just 1 hour.
This is where mitosis happens.
Yeah.
Prophase condenses the chromosomes, metaphase aligns them at the equator, anaphase violently pulls them apart to opposite poles, and telophase reforms 2 distinct nuclei around the separated DNA.
I need to pause here for everyone studying for their TEAS or HESI A2 exams, because the research notes highlight a massive point of confusion regarding the end of M phase. Mitosis and cytokinesis are constantly confused or, treated as the same word.
Oh, constant.
Can you break down the mechanical difference?
Examiners love to test this distinction. Mitosis refers specifically and exclusively to the division of the nucleus and the separation of the chromosomes. It is purely about managing the DNA.
Okay, so that's mitosis.
Right. Cytokinesis is the physical pinching and separation of the actual cytoplasm and the outer cell membrane. It's what ultimately cleaves the single cell into 2 completely separate daughter cells.
So mitosis divides the library, but cytokinesis divides the actual brick-and-mortar building.
That is a great way to think about it. They're completely mechanistically distinct processes.
But they happen around the same time.
They overlap in timing, yeah. Cytokinesis usually starts happening during late mitosis, but they're driven by entirely different molecular machinery. You can actually have mitosis happen without cytokinesis.
Wait, really? What happens then?
It results in one giant cell with multiple nuclei inside it.
Oh, wow. Okay, which brings up the most pressing question of all.
Yeah.
We've talked about the cell taking an inventory in G1 and proofreading DNA in G2, but how does it do that?
The machinery.
Yeah. Who is the factory supervisor enforcing these rules? What actually hits the brakes if the DNA is damaged?
This is where we look at the actual molecular engine driving the entire cycle. The cell possesses this layered biochemical surveillance system made up of checkpoints.
Checkpoints.
There's a strict checkpoint at the G1S transition. That's the R point we discussed. There's another at the G2M transition to ensure the DNA was perfectly copied.
Right.
And there's even a spindle assembly checkpoint during metaphase to make sure every single chromosome is physically tethered to a microscopic tow cable before they are ripped apart.
And the proteins that run these checkpoints have a very specific name: cyclins and cyclin-dependent kinases, or CDKs.
Yes. CDKs are the engines and cyclins are the keys that turn them on.
Okay, so how does that work?
Well, a cyclin-dependent kinase is an enzyme that just floats around the cell in an inactive state. It literally cannot do its job until it binds to a specific cyclin protein.
Let's get into the how here, because calling it an engine feels a bit, abstract. When the cyclin binds to the CDK and turns it on, what does the CDK actually do to push the cell into the next phase?
That requires understanding what a kinase is.
Right.
A kinase is a type of enzyme that acts like a molecular tagger.
A tagger.
Yeah. When the CDK is activated by a cyclin, It changes its physical shape, which allows it to grab onto other proteins in the cell and slap a chemical tag onto them, specifically a phosphate group.
Okay.
By slapping this phosphate group onto a target protein, the CDK radically changes that protein's shape and behavior. It might turn a dormant enzyme on or turn an active one off.
So it's bossing the other proteins around.
Exactly. This cascade of chemical tagging is what physically forces the cell's machinery to advance to the next phase.
Discovering an invisible chemical tagging system feels like it would be an impossible task. Who actually figured this out?
It took decades of painstaking work, which culminated in the 2001 Nobel Prize in Physiology or Medicine.
Oh, wow.
Leland Hartwell used budding yeast in the 1970s, identifying mutant strains that would get physically stuck at specific points in the cycle. Then Paul Nurse discovered the master regulatory kinase, CDK1.
And there was a third winner, right?
Yes, Tim Hunt, who discovered the cyclins. And he did it almost by accident in 1983 while looking at the fertilized eggs of sea urchins.
What I find so incredibly bizarre about Hunt's discovery is the behavior of the cycling protein itself.
It is weird.
Yeah. He noticed a protein on his laboratory gels that would steadily build up in the cell, reach a peak, and then abruptly vanish, entirely in sync with cell division.
Just poof, gone.
Right. It turns out this protein exists solely to be destroyed on a strict schedule. It's like spending your entire weekend meticulously building a massive Lego castle, just to deliberately smash it with a hammer at 5:00 p.m. on Sunday so you know the weekend is officially over.
That is exactly what it's like.
Why would a cell waste so much metabolic energy building something just to destroy it?
It seems like a terrible waste of resources, but that rhythmic rise and violent destruction is actually a stroke of evolutionary brilliance.
How so?
It is exactly what gives the cell cycle its one-way, irreversible direction. Think about it. If the cyclin just hung around, the CDK would stay active, and the cell might try to run the cycle backwards.
Oh.
But by completely annihilating the cyclin, the CDK abruptly shuts off. The bridge is burned. The cell is physically forced to move forward. It can never run the cycle in reverse.
It's a one-way street, enforced by destruction. That is fascinating.
It really is.
But it also leads us to a dark reality. If we have this brilliant system of checkpoints, CDKs, and cyclins, what happens when the factory supervisor gets knocked out? Like, if the cell cycle is a one-way street, what happens when the brakes fail?
When the cell cycle's brakes fail, cells ignore the restriction point. They bypass the DNA proofreading, they divide rapidly, endlessly, and chaotically, piling up on top of one another.
And we have a word for that exact cellular behavior.
We do. Cancer. Fundamentally, cancer is a disease of a broken cell cycle.
So who are the molecular brakes? If cyclins and CDKs are the gas pedal pushing things forward, who are the guardians stopping the car?
We rely on a class of proteins called tumor suppressors. The two most famous are the retinoblastoma protein, known as ARB, and a protein called P53.
Okay, RB and P53.
RB acts as the physical brake pad at the G1 restriction point. But P53 is the true factory supervisor. It is famously known in biology as the guardian of the genome.
The guardian of the genome. How does P53 actually stop the cycle if it senses danger? Does it, like, build a wall?
When P53 detects that the cell's DNA has been damaged, maybe by UV light or a chemical toxin, it acts exactly like a supervisor slamming the emergency stop button on a factory conveyor belt.
Okay.
P53 rapidly accumulates and halts the cell cycle in G1 by inhibiting the CDKs. It locks the doors and refuses to let the cell enter S phase.
So it gives the cell time to fix itself.
Exactly. It gives the repair enzymes time to fix the broken DNA. And if the damage is too catastrophic to be fixed, P53 triggers a process called apoptosis, programmed cell death.
It tells the cell to die.
It orders the cell to gracefully dismantle itself, so the corrupted DNA is never copied.
That is an unbelievable amount of power for one protein, which means if you are born with a defective P53, you essentially don't have an emergency stop button.
Precisely. There is a rare genetic condition called Li-Fraumeni syndrome, where individuals inherit a mutated non-functional copy of the P53 gene.
Wow.
Because they lack that crucial supervisor, they face a massive, near-inevitable lifetime risk of developing multiple types of cancer, often starting in childhood.
That explains inherited cancers. But how does a normal, healthy person with perfectly functioning P53 and ARB still get cancer? How do those molecular bouncers get overpowered?
To understand that, we have to look at the most famous cells in medical history, Helo cells.
Henrietta Lacks.
Yes. These were cervical cancer cells taken without consent from Henrietta Lacks in 1951. They are still dividing in laboratories worldwide today because their cell cycle is completely unhinged.
But she didn't have life from any syndrome.
No, Henrietta Lacks didn't inherit broken breaks. Her cancer was caused by the human papillomavirus, or HPV.
The virus physically broke her cell cycle. How?
The HPV virus produces two highly specialized, almost weaponized proteins called E6 and E7.
E6 and E7.
These viral proteins evolved with one singular purpose: to hunt down and eliminate the host cell's breaks. E7 specifically targets and inactivates the RB protein.
A6.
E6 targets the guardian P53. And the mechanism of how E6 eliminates P53 is incredibly sinister.
Please tell me it doesn't just smash it.
It acts like a biochemical hitman. The E6 viral protein binds to P53 and forces the cell's own machinery to attach a specific chemical tag to it called ubiquitin.
Ubiquitin.
Ubiquitin is essentially the cell's kiss of death. It's a tag that tells the cell, this protein is garbage, take it to the incinerator.
Oh my God.
By forcing P53 to get tagged with ubiquitin, the virus tricks the cell into throwing its own factory supervisor into the cellular wood chipper, a structure called the proteasome.
That completely flips my understanding of virology. The virus isn't just taking over, it's using the cell's own garbage disposal system to murder the security guard.
It really is.
And this is why anyone in the clinical field stresses the importance of the HPV vaccine. It isn't just preventing a mild viral infection. You are literally vaccinating the patient to protect their cellular brake pads. You are keeping the hitmen away from P53.
Exactly. When you lose the brakes, cells divide infinitely. But it's important to realize that infinite division is an anomaly.
Right, because normal cells don't do that.
Normal, healthy cells have strict, unbreakable limits on how many times they can run this cycle.
Wait, I remember reading about a famous experiment from a guy named Alexis Carroll. Didn't he keep the cells of a chicken heart alive and dividing in a petri dish for decades? I thought cells could just keep going if they had enough nutrients.
That chicken heart experiment was one of the most famous and most flawed studies in biology.
Really?
For years, the public and scientists believed Carroll had proven cells were immortal, but he hadn't. It's now widely accepted that his laboratory assistants were accidentally introducing fresh, new chicken cells into the culture every time they fed the tissue with nutrient broth.
Oh my God, they were just accidentally replacing the cells and thinking the originals were living forever.
It is a massive cautionary tale about experimental rigor, because in 1961, an anatomist named Leonard Hayflick proved that normal human cells absolutely do not divide forever. They hit what's now called the Hayflick limit.
The Hayflick limit.
A normal human cell can only run the cell cycle roughly 40 to 60 times before it permanently stops.
What physically starts it, though? Is it a molecular counter?
It's the telomeres. Telomeres are the protective caps on the physical ends of your chromosomes. Think of them like the plastic aglets on the ends of your shoelaces that keep them from fraying.
Okay, the aglets.
Every single time a cell passes through S phase and copies its DNA, the replication machinery physically cannot reach the very tip of the strand. So a tiny piece of the telomere gets snipped off.
Oh, I see.
After 40 to 60 divisions, the plastic aglet is completely worn away. The cell senses that the actual coding DNA is in danger of fraying, and P53 steps in, permanently halting the cell cycle. The cell enters senescence, it ages.
So normal cells age and stop dividing. Cancer cells lose their P53, ignore the telomeres, and divide forever.
Precisely.
Which brings us to the clinical reality of how we treat this on the floor of a hospital. Chemotherapy.
Yes.
I always imagine chemo as this incredibly advanced, targeted, smart bomb. But based on how the cell cycle works, that's not true at all, is it?
No, not at all. Traditional chemotherapy drugs do not know what a cancer cell is. They are completely blind to the disease.
So what do?
Instead, they weaponize the vulnerabilities of the cell cycle itself. Many of these drugs are designed to target and violently disrupt specific phases.
Like S phase.
Exactly. Some chemicals physically cross-link the DNA strands so they can't be pulled apart, triggering catastrophic failure strictly during S phase. Other drugs, like paclitaxel, freeze the microscopic tow cables, the spindle fibers, so the chromosomes can't separate during M phase.
But if the drug is just floating through the bloodstream, attacking any cell that happens to be in S phase or M phase, that explains the brutal collateral damage of chemotherapy.
Precisely. The drugs attack actively cycling cells. Cancer cells cycle rapidly, so they take the brunt of the damage. But what else cycles rapidly in the human body?
Oh, wow. Hair.
Your hair follicles are constantly dividing in S phase to grow hair. The epithelial cells lining your gastrointestinal tract replace themselves every few days. The hematopoietic cells in your bone marrow are relentlessly dividing to create new red and white blood cells.
That is exactly why chemo causes hair loss, why it causes severe nausea and GI distress, why it destroys the immune system.
Yeah.
The hair, the gut, and the bone marrow are just innocent bystanders that happen to be moving too fast through the cell cycle. They get caught in the chemical crossfire. If you don't understand S phase, you simply cannot understand how these drugs work.
It all comes back to the machinery. The cycle governs everything.
It really does. Look, if we quickly recap what we've unpacked here. First, interphase is 90 to 95% of the cycle, not a resting phase.
Number one mistake.
Second, S phase is for DNA replication, thanks to Howard and Pelk. Third, cyclins and CDKs drive the checkpoints.
Nobel winners.
Right. Fourth, mitosis and cytokinesis are distinct. And fifth, the loss of P53 and RB leads to uncontrolled division, like cancer and HeLa cells.
And that is exactly why understanding the nuances between G1, S, G2, and M is so vital. It's why pap smears look for abnormal division patterns. It's why clinical oncologists time medication delivery to maximize S-phase disruption.
The cell cycle is the language of life.
It really is. It changes how you perceive your own body. You aren't just flesh and bone. You are an unfathomably complex factory, running trillions of microscopic restriction checks every single second of your life.
Which leaves us with a final, slightly terrifying thought to mull over for you listening. We talked about how specialized adult cells, like the ones in your brain or your heart, exit the cycle and park in the dormant G0 phase, making their injuries permanent.
Right, they get stuck.
Meanwhile, stem cells retain the special ability to re-enter the cycle at will. There are massive research efforts right now trying to find a chemical signal to force those dormant G0 heart cells back past the restriction point into G1 so they can multiply and heal a heart attack.
The holy grail of cardiology.
But if we actually succeed, if we figure out a drug that successfully overrides the strict G1 checkpoints and forces highly specialized tissue to rapidly divide, would we actually be curing the heart disease? Or would we just be instantly triggering a massive, untreatable cancer by artificially breaking the very brake pads that keep those cells safe?
It's a fine line.
It is a thrilling and terrifying boundary in medicine. It really makes you realize how delicately balanced that invisible rhythmic machinery has to be just to keep us alive. Until next time, keep diving deep.